Hello everyone, welcome back to Toppr.com. So again, on Tuesday we are back with our biology live class. So those who have already attended my last class, we have already discussed about the topic cell cycle and cell division. So first of all, a bit of apologies for being a bit late because of some technical issues, So yeah, today's session will be conducted for the topic photosynthesis in higher plants. So now, in order to conduct this session, what I have thought is we'll be talking about this portion, which is very important, and we'll be talking it in terms of two sections, and maybe it can take three. So what I've decided is in today's session, it won't be too heavy. It will be a very easy session, but very important because we are going to talk about certain experiments which actually led to what we know about photosynthesis right now. Okay. So you understand this is very important, and also we'll be talking about some of the important structures which are essential for this process of photosynthesis. So I hope you are all ready. So once again, subscribe to Toppr. Okay. Now you should also invite your friends. I'm sure from your feedback we have seen that you are being very much benefited by us. So let's start with our today's session. So photosynthesis, right? So it is one of the chapters of your 11th biology. And what is the meaning of this word photosynthesis, right? Let me write it down. Photosynthesis, right? So now this is a simple word, and I'm sure you must be studying this word since your childhood, right? This word means photo. This word has come from the word photon. Okay, and synthesis means to make something. That means that to make something with the help of light. Photons, that is light, correct? So let's see. This photosynthesis process is actually involved in the formation of sugar, let's say food for the plants, correct? We know about this. So this is relevant for all those type of organisms, specifically plants, and there are some also bacteria who can do this process of photosynthesis, correct? Now what are they called? These organisms who can photosynthesize, they are known as autotrophs. Now let's be specific. Do we call them autotrophs? So what's the meaning of this word autotrophs? Autotrophs actually mean someone who can make or prepare its own food. But if we be specific, we can call them as photoautotrophs. So now I'm sure you can guess it. So what does this mean now? It means that the organisms who can make their own food with the help of photon, that means light. So now you tell me, if it becomes certain organisms who can make or prepare its own food but with the help of chemicals, what we'll call them? We'll call them as chemoautotrophs, correct? So these are chemoautotrophs which can obtain their own food or prepare their own food with the help of certain chemicals. So there are certain nitrifying bacteria, there are certain organisms who can do all these things, okay? Now, so we'll talk about the photosynthesis. We'll see the equation of photosynthesis, right? So before that, let's study some of the basic things in photosynthesis. Now I'll tell you a very interesting fact about science actually. So the interesting fact about science, or you will love science if you actually love to study the experiments conducted by various scientists. So here we'll see how this process of photosynthesis, right? We are aware of this photosynthesis process. We have seen the equations that is written on your books. There are certain structures which are given on your books, correct? But have you wondered from where have this come from? Who have invented this? You may not remember the names, but we may be very much interested in knowing how this has been formed, okay? How this has been formulated. So first, in this session, we'll talk about certain experiments conducted by very famous scientists which actually led to the invention of this process of photosynthesis conducted by the plants. Let's see what are they and how are they performed. Now, in your childhood, you must have done certain experiments related to photosynthesis. One of the basic experiment I'm sure you must have done is, which is called, suppose you take pot which has a flower. Okay, let's draw a flower here over. It's not a small tree. Now here we have the leaves, correct? Now what happens? Let's conduct an experiment. Now this experiment was conducted in such a way that certain part of the leaf, this part of the leaf was put inside a glass jar and it was sealed. And what was there inside the glass jar? Inside the glass jar there was KOH. Okay. Now this KOH, this chemical, okay, ये जो chemical है ये actually carbon dioxide को absorb कर लेते हैं. Okay, so KOH जो chemical है, what does it do? It absorbs carbon dioxide. Now this plant was kept in sunlight for about 6 to 7 hours and we were seeing what can happen. So scientists saw what can happen. Okay, now later on it was seen after the conduction of starch analysis. Now you should understand how the scientists actually thought these experiments. So when they try to do the starch analysis and how this is done with the help of iodine. So I'm sure you must have heard about this technique or this test, starch test, which is called iodine test. Here, if you add iodine to starch, it turns what? Blue-black in color. Okay, so it will turn blue-black in color if you add iodine to starch. Okay, right. So I'm getting questions about certain experiments, about specific scientists. Yes. So we are actually going to talk about these experiments slowly and steadily. Okay, don't rush around because this is a chapter which is very important and will actually dive into the experiments and try to understand कि कैसे these theories have been proposed and all these things have been proved. Correct? So what have we studied? That in order to understand the presence of starch, we have to do iodine test. Correct? In which it will turn blue-black in color. Now you try to understand or imagine if I take out this plant and test for iodine. In this leaf, if I pluck this leaf and test for iodine, what will I get? Let's draw this leaf. So if this is the leaf, correct? And now if I do iodine test, if I put iodine solution over here, you know what will happen? This part will turn blue-black. So this will turn blue-black. But here, this won't turn blue-black. Now can you say why this has happened? So what does this mean? This means that the region where blue-black color has arrived, that region has got starch, whereas this region does not have starch. Now let's go back to the experiment once more. So here what we have done, हम लोगों ने KOH use किया, right? This chemical absorbs carbon dioxide. So this container is sealed. अगर ये container sealed है, that means there is no availability of carbon dioxide. Now what does this prove? If there is no carbon dioxide, that means that this part of the leaf could not prepare or could not perform the process of photosynthesis. Hence, starch, which is a product of photosynthesis, cannot be made or cannot be synthesized. Correct? So here, will we get blue-black color? No. Why? Because there is no starch. Starch के absence के वजह से यहाँ पे we do not get a positive test for iodine. So we have proved, so this was the way by which scientists try to prove that carbon dioxide was necessary for the process of photosynthesis. Okay, so now let's talk about different types of leaves. Okay, so we have seen leaves which are variegated. You know what are variegated leaves? You'll see the general color of the leaf is green, but I'm sure you must have seen leaves which has bluish color, bluish green color, yellow colored leaves, and they have different shades in a single leaf. There are white patches also. So these type of leaves are known as the variegated leaves. Now why this has happened? I'm sure you must have heard about the word chlorophyll. Okay, chlorophyll. Now this is the green color of the leaves. So we'll study about this chlorophyll in detail specifically in this chapter. Let's see what happens. So because of the presence of chlorophyll this happens. Now the question is, why the other colors? Because I said that chlorophyll gives the green color. So let's put a question mark here and we'll try to answer as we move on with our chapter. Okay, so we have done or proved that carbon dioxide is necessary for photosynthesis. Now can we move on to the other experiments? Let's see. So now we'll talk about if sunlight is necessary. Can we talk about that? So if we try to talk about sunlight, I'm sure you can do this experiment at your home also. So you can take two plants. Take one plant, keep it for darkness at about for 2 days maybe, and keep one plant in bright sunlight for about 6 to 7 hours. Now take those plants together and do the iodine test. Okay. Now what will you observe? The plant which was kept in dark, जो अंधेरे में था, वो plant पे you would get a negative result. Okay, so for which plant? The plant which was kept in dark. Okay, I hope you can see my handwriting properly. So the plant which is kept in dark shows negative result. That means, will blue black color arise over here? No. Now the other plant which was kept in sunlight, it appears positive for starch test. That means it gives the blue black color, right? So what does this prove? यहाँ से क्या प्रूव होता है? यहाँ से ये प्रूव होता है कि अगर सनलाइट पे रहता है कोई भी प्लांट, if a plant is kept under sunlight, that means that plant is able to perform the process of photosynthesis, and hence starch is prepared. Correct? Starch is the food for plants. That is glucose, a form of starch, right? Basically starch is a form of glucose. Okay, so it's a derivative of glucose. So starch can be formed if a plant is kept under sunlight. So sunlight पे starch बन जाएगा. So we'll get a positive result. Whereas अगर dark पे रख देते हैं, if it is kept in darkness, then we would get a negative result. Why? Because there is no sunlight. So in the absence of light, it cannot perform the process of photosynthesis. Hence it's a negative result. So till now what have we proved? Sunlight is necessary, carbon dioxide is necessary. Now let's talk about certain very interesting early experiments that were performed. Correct? So we'll talk about a scientist whose name is Joseph Priestley. Okay, Joseph Priestley. Let's see what he has done. Now Priestley, if you have heard about this scientist's name, he actually have talked about the element oxygen. He was the first person to talk about or describe oxygen. Correct? Now let's see what he has contributed towards this process of photosynthesis. Correct? Now we'll talk about certain setups. He used very simple type of setup. He used a jar, glass jar, and call it as a bell jar. Okay, so this is a jar. Now inside this jar he has kept a candle. So here's a candle, and also there's a mouse, you can say. So let's draw it like this. Okay, now what was observed, he lit this candle. This candle was lit. After some time he saw that this mouse actually died when this candle was put out. Okay, slowly and steadily, since this is actually airtight chamber, air के कोई भी रास्ता नहीं है कि इसके अंदर आ जाए. So airtight है, इसका मतलब है इसके अंदर जो भी एयर है वो यूज करके ये बर्न हो रहा था. कुछ देर बाद देखा गया कि ये लाइट बंद हो जाता है, यानी कि this fire goes off. After this fire goes off, till the fire was on, it was seen that the mouse was alive. But after this fire goes off, this candle goes off, it was seen that after some time this mouse, it dies. It suffocates, and then after some time it dies. So what does this prove? From this experiment, now you think about how actually Priestley he himself thought. Now think, If you have yourself done this experiment, now what would you have concluded from this experiment? I would have concluded like this, that since I have started with the setup in which the candle was lit, that means that here there was certain air which was helping the candle to burn, and also this mice to stay alive. But slowly and steadily, the burning of this candle as well as the mice, this animal, it has perhaps given out certain poisonous gases which started accumulating here and which cannot escape, because of which it died. Can this be a conclusion? If yes, actually Priestley, he himself also did or concluded the same thing. Now he thought, let's modify this experiment a bit. So what he did was, he took the same setup. Here we have this jar, we have the candle also. Saw the candle, correct? Now the addition was, he put a mint plant over here. A mint plant was put, okay? And there was also the mice which was present. So you know, this mice, they are very poor creatures, because we perform various types of experiments with these mice, okay? So we have to actually sacrifice a lot of mice because of our experiments. Now let's see what was observed. When he did this setup, he just modified here. What was the modification? He added a mint plant under this setup. He found that the light did not go off, the candle did not go off, as well as the mice was alive. Let's repeat this experiment. So what was observed, that if just a small addition was done to our previous setup, what was done was, a mint plant was kept over here. So because of this mint plant, it was seen the observation actually changed. The mice did not die, the candle was also not put off, which was seen initially. Now from here, what can you conclude? I actually conclude from here that this burning of candle and this animal was using, these two were using something which is present inside the glass jar. Now with the use of plant, with the addition of plant, the plant actually replenished or gave back that same thing which was being used by the candle and the mice, correct? Let's repeat once. So here what we have seen, that the candle is using something, we concluded that the candle is using something, the mice is using something. We don't know what is that, but since it's, when it gets over, because this is a glass sealed container, so when it gets over, the candle goes off, the mice dies. But the same thing was done over here, but with the addition of plant, it means the things which were being used by the plant was used by the candle and the animals, that was being put back or replenished by the mint plant. So from here actually it was concluded that there is some relation between the plants and the animals, that plants give out something which is being used by the animals, or it helps in burning, and the vice versa. The animals or the candle, it actually gives out something because of burning, and the animals give out something which is poisonous, which is taken back by the plants, right? So this was the relation set up by Priestley. Now I can give you a very interesting question over here. You can think it about the question, the answer of this question at your home, and you can surely get back to us with your answers. So the question is, if this light goes off in some way after a long period of time, now can you tell me how would have Priestley lit this candle once again, maintaining the same condition? So the question is, अगर ये लाइट ऑफ हो गया एक टाइम पे, अगेन उसको बर्न करना पड़ेगा, उसको जलाना पड़ेगा, दैट मीन्स आई हैव टू लाइट दिस कैंडल वंस मोर. How this can be done without disturbing the setup of the candle? To understand, now the question is, why do we need to do this? We need to do this to understand if something has changed or not, if the light goes off, what was used or what was not used. So think about this question, you may arrive at very interesting answers. Okay, so let's move on with our experiments now. So along with this, we have talked about the dark experiment, okay? The half-life experiment we have discussed previously only, in which we have used KOH and proved that carbon dioxide is necessary for the process of photosynthesis, correct? So in between, if you have questions, if you have doubts, please put up your questions so that I can answer your doubts or solve your doubts, okay? So we'll be discussing along with your doubts also. If you have doubts, please put up. Okay, so we were talking about certain experiments. We have talked about certain important experiments. Let's move on. Now, so from here, these experiments were now based on these conclusions of the experiments we have discussed so far. Certain scientists, they advanced these type of experiments. So one of the scientists, actually, what he did was, Engelmann, he's called the T.W. Engelmann. What he did was, he used cladophora. Okay, cladophora, this is basically a green algae. Now, this can photosynthesize. Try to understand. He used a culture in which cladophora is a green algae and it can photosynthesize. And he used bacteria. Okay, bacteria which are aerobic. Now, you have to understand this. What does this mean? This means that this type of bacteria, for their survival, they require oxygen. Aerobic means उनको अपने survival के लिए, बचने के लिए, they require oxygen, correct? So they are known as aerobic organisms. Now, what he did was, he cultured this cladophora, which is an algae, and he found that the areas in the culture where this cladophora was growing shows the growth of this bacteria. That means, let's repeat, let me draw it here. If this is our sample space in which we are going to see our experiment, observe our experiment, let's suppose this blue shaded area, the cladophora or the algae. These are the, if you can see what I am drawing, if you cannot see the color also, no problem. So the color which I am drawing now, suppose these two areas are the algae. Let's write it down. Correct? Cladophora. Now, he actually inoculated bacteria also. Now he found that this bacteria actually grew in these areas. Okay, I hope you can see it. Yes, it is visible. So these are the areas where bacteria grew. But all these areas did not show the growth of bacteria. Now this was a controlled culture in which there was no external source of oxygen. So what does this mean? This means that the oxygen which was produced, because we know that aerobic bacteria are the bacteria which will require oxygen for its survival. So this oxygen which was produced by the algae was used by the bacteria for their survival and they grew. So उनका नंबर बढ़ गया इन algae के पास ही, क्योंकि algae actually oxygen produce कर रहा था. और in these areas, okay, here oxygen was not being produced. So what happened? The bacteria could not grow because these are not anaerobic bacteria. These are aerobic bacteria. So it requires oxygen for its survival. Correct? So you understood how it was proved again that oxygen is the material which is used or which is essential for, which is produced actually as a result of the process of photosynthesis. In fact, at this point of time, the word photosynthesis was not coined. It was also not known that this is something happening in photosynthesis. Okay. It's just that certain elements or the raw materials of this photosynthesis and the products of the photosynthesis were being discovered. Correct? हमने देखा है कि कौन-कौन से raw materials use हो रहे हैं plant के survival के लिए, और we try to prove it, and also we try to prove what are the products which are formed with the process of photosynthesis. Correct? So we have proved oxygen is one of the product of photosynthesis. Correct? Now let's move on with other experiments. So let's see if we have any questions. Okay. Yeah. So I got a question about cladophora. Yeah, we have already discussed, right? We are moving on with your doubts. Yeah. So now, interesting again, our next advanced process. Now, what happened? I'm sure you heard about a prism, correct? very common word in your physics. You have done many experiments in physics. In fact, you have used it as a toy also, correct? Now, think about what happened when you put a torchlight in this prism. Can you tell me what happens? What happens is, you'll see from the other part of this prism, there are certain rays of light, and if you count it properly, there will be 7 such rays. I'm sure you know this. This is with jaw. Violet, indigo, blue, green, yellow, orange, red. So this is a sequence in which you are going to get these colors. But the color of the light from the torch was what? It was white. So we can say that this white light is composed of 7 different colors, which is following this order with jaw, correct? And if you pass it through a prism, there is a very interesting physics around this. You can study this in your physics classes. So here, this prism actually helps in splitting the colors, splitting the white light into its component colors, correct? You understood this? Now this experiment was done to understand, so if this white light, the sunlight which we are getting is white light, right? Now, can we tell which color specifically from this VIBGYOR is actually helping in the process of photosynthesis or preparation of the food for the plants? What was done is, the same type of experiments was carried out by the prism, in which specifically certain areas, certain plants were illuminated. Illuminated means they were lighted up with these parts of the light and not the white light together. So you understand, the parts of the light were used to illuminate the organisms which can actually photosynthesize. Now you know what was interestingly observed? It was observed that this blue and this red are the regions which, when illuminated by these colored rays, that is blue colored light and red colored light, then only the organisms could perform the process of photosynthesis. So from here, what do we conclude? This experiment concludes that the light blue, the range between blue and red are the best areas, best range of light which actually helps in the process of photosynthesis, correct? But I'll tell you an interesting fact. There are certain other pigments also. So we'll talk about it. There are certain various other pigments which help in the process of photosynthesis, but they can perform the process of photosynthesis with the help of other colored lights also. So this is a bit of advanced thing which you do not require at this point of time, but yes, you should know the best amount of photosynthesis can be done with blue, red light ranges, correct? So these were the certain experiments. Now one of the important experiment. So from here we know that water plus carbon dioxide gives what? Glucose, C6H12O6 plus what? Oxygen. We know this is the process of, this is the equation of photosynthesis, correct? Someone was asking about the equation of photosynthesis. So this is the basic equation of photosynthesis. You have to balance it, correct? Now what happens is, with this equation, so okay, yeah, fine. So with this equation, what happens is, we'll see this oxygen is a product of this process of photosynthesis. We have already proved it. Now there are 2 raw materials, water, carbon dioxide. Both of these contain oxygen. Now the question is, so from where is this oxygen coming? Is this oxygen which is released, this oxygen is released out, correct? And this is the oxygen which we inspire or which we inhale. This oxygen is coming from carbon dioxide or it is coming from water. So what do you think? This was proved by various types of experiment, and I'll be talking about a modern experiment in which there was use of isotopes. Isotopes, these are known as radioactive. You know, these are certain materials which are radioactive. Now radioactive isotopes of oxygen were used. So what was done, there was a very interesting experiment which was done is, so in one of the reaction, water, in construction of water, radioactive oxygen was used, and in the other one radioactive carbon dioxide was used. So surprisingly, it was observed that initially it was thought that from carbon dioxide oxygen is coming, but interestingly it was seen that when water was labeled, okay, so in this radioactive isotope this was labeled, so it was observed the oxygen which is coming out after photosynthesis, this is labeled. But if we label just carbon dioxide, oxygen of carbon dioxide, and see the oxygen, it does not come labeled. Okay, so what does it prove? It proves that the oxygen is coming from water and not the carbon dioxide. Correct? So yes, we are almost done with the important experiments. Now we can talk about certain features. One of the important structure which is essential for you to understand the process of photosynthesis. So can you guess what structure we are going to talk about? We are going to talk about a structure. Let's draw it so that we can understand it properly. And this diagram is very essential because this diagram may be asked in your school examinations. So this is what? Chloroplast. Okay, the portions in the plant which is present in the mesophyll cells. Okay, mesophyll cells of the plant. So you should remember it. It may be asked in your MCQs. Where are these chloroplasts found in plant cells? These are found mostly in the mesophyll cells of the plants. Now let's see in detail what is the structure of this chloroplast. So this chloroplast is, first of all, you should know it's a double membrane bound structure. Again, a very interesting and one of the most common questions asked in your examinations is name some double membrane bound organelles present in plant cells or animal cells. So chloroplast is one of the organelle which is double membrane bound. Correct? So this is the outer, correct? I hope you can see it, and this one is the inner membrane. Okay, so I have got a question about radioactive oxygen. Someone wants to answer or know about the radioactive experiment once more. Okay, so no problem, we can quickly discuss that experiment once more. So let's take back the sheet once more. Okay, so we have got this sheet where we have already written the experiment or the equation of photosynthesis, correct? So here what we have seen, that there is water, there is carbon dioxide, it results into the production of glucose as well as it results in the production of oxygen. Now what was the objective of this experiment? The objective of this experiment was that we want to prove from where this oxygen is coming. Now what are the probabilities of oxygen coming from? It can come from water, it can come from oxygen, the carbon dioxide, because both of them has oxygen as their element, correct? Now how to prove? Now there we have used radioactive isotopes. Oxygen, it is generally not radioactive. Radioactive is something where these chemicals, these elements, if they are radioactive, they can be detected easily. So there we have radioactive detectors and we can detect if these materials are radioactive or not. So I can tell you in very simple languages that it's basically labeling. For example, if you stand in a crowd, in your school, if you stand in a crowd and no one is wearing a cap, but I give you a red color cap, and if I'm standing at the stage, if I want to find where you are, I can easily locate the cap, right? Because if there are thousands of students, I cannot locate you. But if that cap is there, that is basically, you can think it as or relate it as a radioactive labeling. So that is basically labeling. So what we have done is labeling of the oxygen, right? Now let's divide this experiment into two halves. In first half we have used, so we will use all the raw materials, of course, because or else the glucose would not be formed. So in one case we have used oxygen in water which is labeled. So here in the first case, this one will not be labeled, correct? You understood? So in the first case, oxygen of water is labeled and oxygen of carbon dioxide is not labeled. In the second case, oxygen of carbon dioxide is labeled but oxygen of water is not labeled. What was found in the first case, the product of this photosynthesis, here we found labeling in oxygen, whereas in the second case where carbon dioxide was labeled, but not water. Here we did not get oxygen. So what does this mean? From where this oxygen came from? It cannot come from anywhere, right? It has to come from the raw materials. So it came from the water, correct? So this was the process by which it was proved that water is the source of oxygen and not carbon dioxide. I hope you have understood this. So let's continue with the structure of chloroplast. We have the inner and the outer, inner membrane and the outer membrane. And what are the other important areas? So this, inside this, between these two layers is a fluid filled layer. Inside this chloroplast, this whole space you are seeing is covered by a fluid which is, and it is termed as stroma. Okay, this stroma, or it can also be referred to as the matrix. This stroma actually is the house of various components. For example, it is the house of RNA. So you must know that these chloroplasts, they have their own DNA also. Okay, chloroplasts, they have the DNA and it is present in the stroma. It has got RNA, it has got various proteins, it has got enzymes which can help in the process of carbon dioxide assimilation. So you'll see where this carbon dioxide is used. So what do we mean by assimilation? Assimilation is basically the process जिससे carbon dioxide use होते हैं. Okay, so हम देखेंगे कि this process of assimilation of carbon dioxide happens in stroma. So can we write it down? There are DNA, we have RNA, we have enzymes, correct? We have various proteins, we have carbon dioxide assimilation, okay, and also we have certain ribosomes over here because it helps in the process of protein synthesis. There are fat globules, there are starch granules. So all these components are majorly present in the stroma. So this is the fluid medium. Now let's move inside. So inside the structure, there are certain disc-shaped structures. Okay, now you can see these disc-shaped structures. Each of these disc-shaped structure is called as thylakoid. Okay, thylakoids, correct? You got it? So I have drawn 4 such disc-shaped structures. One of each of them are called thylakoids, correct? Now this stack of thylakoids, you see there are 4 which has done a stack. This stacked thylakoid, it is called as what? It's called as the grana. Correct, you got it? So I can repeat it once more. Each of these structures, disc-shaped structures, these are known as thylakoids. They are membrane bound structures, and these thylakoids together they form the stack which is called the grana. So grana is stack of thylakoids. Correct, you got it? So you'll see how these structures are important. So these type of stacks are actually spread all over the chloroplast. Okay, so I'm not doing fully, so I've drawn some. Now these stacks, they are interlinked, interconnected by certain structures. Similar stack-like structures, they're connected, they're interconnected with each other. Now these portions, okay, these portions are called as stroma lamellae. Okay, I hope you can see it, like it's called stroma lamellae. Now these thylakoid, they are also known as lamellae. Okay, right? Or it is also called the fret membranes. So thylakoid or lamellae or the fret membranes. So now how would you remember? You just have to remember that the stacks, each of this disc, they are known as thylakoid. Okay, group of disc, they are known as grana. So I can make you understand how you will remember. So there are 2 words. One is a bigger word, that is thylakoid. One is a small word, that is grana. So the actual relation is reverse. Stack, that means the bigger part is actually the name which is smaller in size, that is grana, whereas the smaller part, which is each of this disc, has a bigger name which is thylakoid. I hope it can help you to remember the names, correct? Now, so these are thylakoids, they are also known as lamellae, fret membranes. Now, since these are connected by these areas and they are in the stroma, hence they are also known as stroma lamellae. Thylakoids together they make grana, and these interconnections they are known as stroma lamellae. So inside these we have like starch granules, there are ribosomes, there are various fats we have studied, we have talked about this. So this is the basic structure of the chloroplast, and the actual process of photosynthesis happens in this portion, right? We'll see how this happens, and very interestingly this happens in the membranes of these thylakoids, correct? We'll see. So you should just take a look in this structure properly, and if you have any questions you can just come up with that. So because we have to be clear about this structure, then only we can understand how the next process of photosynthesis happens. I'm sure this is clear. Now we can move on to the next part. So I'll quickly cover some of the pigments, right? So we have heard about chlorophyll. Now this chlorophyll is just not chlorophyll because there are certain types. Chlorophyll A, B, C. So there are most often the three types of chlorophyll that are present, right? So these chlorophyll, now which is the chlorophyll which actually which is the chlorophyll which helps in the process of photosynthesis maximum? Okay, it was observed that chlorophyll A is the part which actually helps in the process of photosynthesis. It was proved by various types of experiments. We did not know these experiments right now. So in addition to that, there are certain pigments called xanthophylls. There can be carotenoids, right? Now if you just recall, when we started our discussion we had talked about a variegated leaf, and we had put a question mark, why these different kinds of color? These different kinds of color actually happens because of the presence of these types of pigments. Chlorophyll, it gives the green color. Okay, chlorophyll A specifically gives the green color. Xanthophylls, they give yellow type of color. Carotenoids, it gives yellow to yellowish orange colors. So these type of pigments, they actually help in the coloration of the leaves. Now you understand, based on what type of pigment is present, the color of the leaf is there. Now you can ask a question, if chlorophyll helps in the process of photosynthesis, why do we need these type of pigments? Now we will actually understand about this question later on, but we can just tell it very easily. Chlorophyll A is the center, it's a boss. Okay, it helps in the process of photosynthesis. Now xanthophylls, carotenoids and all these pigments, they are known as accessory pigments, correct? You saw it. Accessory pigments means they do not directly take part in photosynthesis process or photo reaction, but they help chlorophyll A to harvest sunlight. You know what happens? So I'll state it very quickly. Chlorophyll A actually harvests sunlight, but if in every reaction center there is one chlorophyll A, but there are hundreds of these accessory pigments. So these accessory pigments actually help in extracting sunlight, and they pass on this energy to chlorophyll A so that the reaction can happen. And also it also helps to reduce the photo oxidation of chlorophyll A, because you understand, if there is too much exposure of sunlight, it can actually damage this chlorophyll A. So it helps in this way, and they are known as, and hence they are called as, accessory pigments. So you understand, there was an experiment which actually shows that chlorophyll A has shown the best amount of photosynthesis which can occur in the presence of blue and red light. Okay, we have already seen this experiment with the help of the prism. Now, so for today's class we'll talk about this, and we'll just talk about two important systems: photosystem 1, photosystem 2. So PS1, that is photosystem 1 and photosystem 2, are the important systems which are actually helping in the process of photosynthesis. I'll explain you what happens. So I've talked about a word, that is reaction center. So this is chlorophyll A. and here we have the accessory pigments. Now these accessory pigments help in capturing sunlight and provides it to chlorophyll. Chlorophyll helps in the photosystem reaction. Okay. Now this photosystem reaction, this PS1 is called as light harvesting system. Light harvesting system, because why? I hope you can understand, because it helps in the harvesting of the light. Now we'll see how this photosystem actually helps in the process of photosynthesis. Okay, so for today's video we have, what have we covered? Let's quickly summarize. So we have talked about various experiments which proves the process of photosynthesis. We have talked about various types of raw materials which were there. हम लोगों ने देखा कि कैसे, how scientists actually proved that these are the raw materials which are essential for the process of photosynthesis. Also, we have discussed about each of these experiments. We have seen how through radioactive experiments oxygen was proved as a product which is coming from water and not from carbon dioxide. And you should remember this, because this is one of the most favorite questions of examiners. It may be asked in your examinations. So oxygen coming from water, it was proved with the help of radioactive oxygen. We have very importantly talked about the structure of the chloroplast. Okay, so this structure is very important. I'm telling you repeatedly, please try to understand this structure, because later on in our coming sessions we'll be discussing about the photosystems which is happening in this membranes. You'll see in this thylakoid membranes the systems which we have talked about, that is photosystem 1 and photosystem 2, they will be taking place in this thylakoid membrane. So we'll talk about photosystem 1, photosystem 2. We'll see how this part is happening, this photosystem. We'll see how energy helps in electron emission. Okay, electron is emitted and this reaction is being carried out in this process of photosynthesis. Okay, so in this session we are not covering much because it will be very difficult for you to understand everything together. So as of now we'll end it here. We'll understand the structure, and in the next session we'll be talking about actually the important sections. Okay, so again, before we end it, please post your questions so that I can quickly answer it, right? Any doubts, we can try to solve it. So what, let me see if we have any questions. Okay, so yeah, I have got a question. Xanthophyll color, okay, it's mostly yellow. Xanthophyll is yellow. Actually you cannot say yellow, it's a bit of yellow orange tinge. It's mostly towards yellow but orange, because if you extract actually xanthophyll, there has been many experiments carried out by xanthophylls. It's mostly yellowish in color, but there's orange tinge in it also. Mostly xanthophylls and carotenoids has more of orange tinge. Correct? So again, I'm repeating, please post your questions. If you have any doubts, we can quickly solve it. And in the meantime, when you post your questions, we can quickly revise what we have already learned. We have talked about certain experiments with the help of we have talked about certain experiments in which we have seen how the raw materials were proved. We have seen with the help of the KOH experiment, in which it was proved that carbon dioxide is necessary for the process of photosynthesis. Then we have seen with the help of Priestley, we tried to associate how the experiment have shown that plants actually help in renewing certain things which are being used up by a burning candle and an animal, right? And here I have asked you a question also, how Priestley could have lighted the candle again. Try to think it and get back with your answers, and trust me, it will be very interesting answers. You can think about it. Okay, so can we do a quick quiz about what we have studied? Okay, so I'll be asking you very easy questions. Let's see if you have understood or not. Correct? Now, very easy questions. You can post your answers also. Let's see if you can get the answers. You can have discussions within yourself also. So if I ask you that at your home, what can be the setup, or what is the compound which you will use to eliminate carbon dioxide? What do you think? We have already discussed in an experiment. So it's a very easy answer. So I'm sure you people have recognized it. So it's KOH, right? So we have done in which experiment? Can you tell me which experiment we have seen this KOH use? It was called as half leaf experiment. Okay, half leaf. Correct. Now this is very important because it may be asked to describe what this half leaf experiment is. Okay. Now let's see if I can ask you any other question. So of course I have asked you one question, that how Priestley could have lighted that candle inside in order to maintain that constant setup. And also in addition to that, so again can you tell me why carotenoid and xanthophyll will be called as accessory pigments and not the core compound of the reaction? Again, I'll repeat the question. Why xanthophyll and carotenoids are the accessory pigments and not the core one? Quickly think about it. Yes. So I am telling you the answer. So these are accessory pigments. Why? We have discussed because these do not take part in the process of photosynthesis, but they help in the process of photosynthesis. How? They have tried to extract the sunlight and give it to whom? Who is the hero of this center process of this reaction? Who is the hero? Who is the boss? That is chlorophyll. And can you name which particular chlorophyll? We have talked about 3 chlorophylls. Chlorophyll A, B and C. Can you name it quickly? I'm sure you can name it. It's chlorophyll A, right? Chlorophyll A is the boss or the center core of the reaction center. Okay, so we have already talked it. So this is the chlorophyll A. Okay, so chlorophyll A actually helps, and these are the accessory pigments. And what are the 2 types of systems, photosystems, that we have already seen? Don't see over here because it's already written. There are 2 types of systems. What are they called? They're called as photosystems. How many types are there? There are 2 types of photosystems. So we are going to talk about these photosystems, and interestingly, let me just tell you what we are going to discuss in the coming session. We'll talk about dark reaction. We'll see what are the process of dark reaction, what are light reactions. So light reaction and dark reaction. Okay, so there's a very important miscommunication or misconfusion you have over here, is that light reaction is that reaction which requires light, perfect, but dark reaction is not that reaction which requires darkness. But actually that is the reaction, you'll see, it does not require light, hence it is known as the dark reaction. Okay, I'm sure you must have heard it in your books. So we'll talk about light reaction, we'll talk about dark reaction, we'll talk about a very important scheme that is known as the Z scheme. We'll see how photosystem 1 and photosystem 2 is interconnected by the Z scheme, where the energy is produced. So we are going to talk about all these in detail in our coming session. So again, if you have any doubts, keep posting us. We'll surely try to answer your doubts, right? And share your views and comments with us, correct? Also try to invite your friends so that they can get benefited by our sessions. We are conducting these type of sessions every day. Okay, most of the weeks we are conducting. Last week we have conducted biology seminar, biology live class for cell cycle and cell division. I hope you have understood that. Today we are carrying out with our topic photosynthesis higher plants. Next week we'll continue this topic. Also we have seminars or live classes for physics, chemistry, also mathematics also. So keep watching them, get benefited from them, get your friends benefited from them, and help them so that they can also get benefited from us. Okay, subscribe to our YouTube channel. I hope you have been clear about this topic, photosynthesis. See you again.